In this video, we'll look at how to generate figures using Pymol for your assignments. So let's look at the first example. So let's say you get a question like explain what is a hydrogen bond and generate an image to show this type of bond. First, let's tackle the theory part. So avoid just giving a short description of a hydrogen bond, but rather provide the definition of a hydrogen bond and then go into more detail. Explain what is this type of interaction, the type of atoms involved and their charges. Are there certain types of elements typically that form hydrogen bonds? What are the strengths of these bonds compared to other types of bonds? As well as what is your typical distances of hydrogen bonds. So next, I'll show you figures that you can generate to indicate hydrogen bonds. But first, let's look at what to avoid. So in this first figure, there's a lot going on. We have the cartoon representation and the line representation, and we have these dashed lines, but we can't even clearly see between which atoms are being connected. So avoid having too much clutter in your image because then you can't clearly see the type of bond you're trying to indicate. This is another example. So it's a bit better than the previous one, but this DNA base is not fully shown in the image. So we can't really make out what is going on here. We don't want to go on assumptions. We want to go on clear images to show a person who is viewing our work to clearly show them what we would like to show them. This figure is much better than the previous two, so we can clearly see the two DNA bases. They are nice and neatly shown. And here we can see the hydrogen bonds and between which atoms they formed. However, the numbers are a bit obscured by these dashed lines. So we can then just drag the label above the dashed lines. So this is the type of figure you should aim to generate for your assignments. So now that we have the theory and the types of figures, now let's look at the figure legends. So avoid just saying image above shows hydrogen bonds. And this really is some of the answers that I have gotten on students' assignments. And this does not get you marks. What gets you marks is detail. So say this is figure 1 and then mention hydrogen bonds are observed and indicated as the dashed lines between the two DNA bases, guanine left and cytosine right. If you want, you can indicate these on the figure itself. You can create, for example, a bracket and say guanine, create a bracket, say cytosine, but you have to do this after creating the final figure. You can add these labels in PowerPoint afterwards. So if you don't include it on the figure, make sure it's included in the figure legend. And then also mention what are these numbers that you are showing. Mention that it is the distance of the hydrogen bond and also provide the unit. If you do not provide the units next to the distance in the figure itself, make sure to include it in the figure legend. And also mention which atoms are shown and what their colors are. If you show this to someone, they will not necessarily know that the red represents oxygen and the blue represents nitrogen. So you have to provide a reader with this types of detail. So let's look at another example. Let's say a question asks you to generate an image for this model and to only include essential items with a detailed figure legend. So let's tackle the theory part first. Avoid just giving the bare minimum of an answer, but rather go into the details. Provide the protein name, not just the PDB code. And you can get this protein name from the protein data bank. Mention what is the function of this protein, why is it important, and also what type of ligands or extra ions or molecules does it require to perform its function. So now let's look at some figures. This figure had the least amount of effort put into it. It is the model only shown in cartoon representation. So avoid doing the bare minimum. Rather provide an image more in the lines of this. It's not perfect yet, but it's better. So the protein is shown in the surface representation, and we can now more clearly see that the DNA is interacting with this protein. This figure is already much better than the previous two. We can see that the DNA is shown as its bases, and we can see how it is interacting in these parts. But what is much better is to actually show the ligand as well in the figure. So now let's look at the figure legend. Avoid the bare minimum. Rather say that this is figure 2. Give a short description of what's happening in the figure. So what type of protein it is and how is it interacting with the DNA, or what type of function is it trying to perform with the DNA. Explain the different representations used. So say that the protein is shown in a surface representation in a light gray color, and then also explain the colors used for the DNA and in this example the ligand. So with the color selection, the DNA has the default coloration, which is the green carbons, where the ligand has a magenta carbon. So again, if you do not wish to add those types of details in the figure legend, make sure to include them on the figure. Again, the more detail you provide, the better. Best of luck.